My name is Julia, and today I'm going to tell you a little bit about the finale project of this fellowship for me called Watch Agent Run. I swear I didn't bribe it. I've called the repo Watch Agent Run, and the goal here is to make traces informative for scientists. Having spent 3 months doing this fellowship, I now firmly believe that the biggest problem, at least that I'm facing, with using AI agents to do anything that I care about, where accuracy is important for science, is that the opaque nature of these outputs just completely impede my ability to trust them. I put a prompt in, and then something happens, and then I get some sort of result, and I feel like this. I'm looking at it, maybe, like, what happened? And I am not the only one, as you've seen the questions just tonight, but if you run a hackathon, you also get to poll hundreds of people about what they think the biggest problem is for AI-driven science. So this is an SF event we're hosting next week, and the question was, what's the biggest problem in agentic science? A brief sampling of our results. We should be spending 1,000 times more time on building verification loops. We have almost no way to evaluate an agent on genuine scientific discovery. Reliability, accuracy, determinism, memory, observability, making agents more trustworthy. I'll stop the barrage, but you get the point. At a completely different event I was at last week in Boston, similar kind of crowd, but totally independent, completely different part of the country. The registration question for this one was, what would it take to trust your agent's output? Again, representative examples. Show me the work. Prove it to me from the outside. No hallucinations. The data source plus every operation between it and the output. Map the task where the model starts to fail and draw the line. So over and over and over again, what you see, the common theme here is people want to believe. These agents seem miraculous. The output is incredible, but how on earth do I know if I can trust it? I'm proposing tonight, and thank you again to the people who wrote this at the AI runtime, one approach for building trust in an agentic system, I think, lies somewhere in the middle between 3 different models of what's going on. One, what should happen, what you actually intended for your agent to do as communicated by a specification or a prompt. Number 2 is what supposedly happened. That's the agent self-report, right? The little summary that you get. And then there's this one, what actually happened as reported by the append-only logs. And so my working definition right now of trust in the AI agent context is that trust is making this checkable against this. What supposedly happened, checkable against some sort of version of reality. And that was the focus of my project, trying to build the infrastructure to do that. A little bit about how it works. Yeah, so the problem is that just looking, I don't know about you, I can't read this. That's what an agent log looks like. It looks like the matrix. It's just sort of, it's a JSON. Maybe if you're a computer, you can read it, but I can't make anything useful out of this. So I built deterministic readouts. Taking this as the source, I made it, one script converts this into an input-output table. Another one that we're going to show the results of in a little bit converts this into a picture that you can sort of read what happened based on the color coding. And then a third one that I'm not going to talk about tonight, but is also in the repo if you're interested, is kind of a provenance reporter for sort of the hero artifact generation, right? Like tracing the specific calls that were related to the generation of the artifact you actually care about. And then, importantly, at least for my own usage, I put all of this into an easy wrapper where you can do initialize run, setting up the infrastructure, and finish run to pull the trace and run the analyses automatically. If it didn't come in an easy package, I wouldn't be able to use it, so I don't expect anyone else to do it either. And I'm going to show you now what that actually looks like. So just as a run, you start out in new run. It sets up the run for you. You can have a container now to write down exactly what prompt you use to set this agent going off. In this case, I put this in as a prompt. We're going to see dangerously skip permissions. I did it as copy-paste, so there's no variance between what the spec was and what started out the run. And then you've all seen this happen. Cloud Code is going to do some stuff, and it's going to keep doing stuff. It's loading my paperclip skill. We like paperclip. Doing all the things, skipping over 10 minutes of Cloud Code, doing whatever it does. It made something happen. We now have results in there that weren't in there before. And then here you have infra, finish, run, and match the name. And now you have the actual trace file, this uninterpretable matrix of what really happened. You have the picture readout, and then you have sort of a table of like input, output, what actually happened. And so to make that a little bit less abstract, I'm going to spend the rest of my time showing you a specific example that I've worked through using this package. So for the biologists in the room, myself being one of them, the simple task that I decided to assign my agent to test out this infrastructure was to extract macrophage differentiation protocols across papers. For the uninitiated, macrophages are immune cells that live all over your body. They're kind of a first line of innate immune defense. They do very important things. And to study them in the lab, you have to take a mouse, you have to get their bone marrow, you have to pull the cells out, you have to plate them, and then you have to kind of let them go for a while. But the details are quite important. Specifically, if you're going to have a specific protocol, you want to know what kind of mouse, which growth factor, how long, all these things that are reported together, usually in prose, in the method sections of these tables. And with all the love in the world to my favorite postdoc in my lab who just told me this is how we differentiate macrophages in our lab, what I wanted to know is like, is that the field standard, right? Is that the only way to do this? If I could read 200 papers and explore different approaches, like what's the variation, right? How is this varying as real people actually do it? And so I seeded a couple of different approaches, basically just extract protocols, do them, but also write it in a column with a little bit more description about what counts as a protocol. The actual results, it's just a table, and then this one is one table. Second run, totally different table. Some of the dates have been converted to July, so there's a little bit of data corruption in there. But the point being, it's kind of a different table than the first one. And I'm like, well, how did that come to be? And what process produced that? And so this is the picture view. I've processed those traces into setup. Read is in blue. And read, where a specific line address was returned in the command, is this blue plus the black. And at 8 papers, it didn't really matter all that much. All 3 prompting approaches were basically the same. It kind of read everything and reported out, okay. But at 200 papers, when you have a context window difference, suddenly the approach that you put in the prompt actually started to matter. You have one pattern for the bare minimum prompt. You have a completely different pattern, as it turns out, when all you do is tell it to make a table. And then you have a different prompt still when you're more specific about the rules. And the reason that this approach, that this matters at all, is thinking about the accuracy of what came back. So, who cares? At the n equals 8 level, nobody, really. All the agents claimed to cover all 8 papers. They all actually covered all 8 papers. There were 0 false positives. The accuracy was basically the same. In the 200 papers, though, you have different kind of approaches. I summarize them as like filter and then reread. They all claim 200 coverage, but actually only the last one got there. None of them, shockingly, had any meaningful amounts of hallucination. This is a disagreement between me and my agents about what counts as a mouse. But the number of true positives, as scored in a subset of 20 of the 200 papers, like I found 16 reading them, so I know that's at least that many. This one recovered half of them, this one recovered 3/4 of them. And the different approaches led to meaningfully different quality metrics as the results of this meta-analysis. And so, in conclusion, if I'm going to leave you with one thought tonight, what you can see, you can begin to trust. That's the thesis that I'm developing and working on as I exit this fellowship. And what I'm hoping for in this room, if you're a researcher and your team working on agentic work, life sciences, work within the life sciences, tell me what you wish you could see well enough to inspect. I'm not sure that my readout matches what you're trying to do, but I think this is an approach worth developing, and I think it'd be very, very useful to hear from a whole range of scientists what they actually care enough about to measure in this way. Thank you so much.